today we are going to discuss about the chemical regulation of respiration, right? So mainly our respiration is regulated by mainly three mechanisms. One is the neural regulation or the neural control. The second one is known as the chemical control of respiration, and the third one is known as the non-chemical regulation of respiration, right? So the non-chemical control includes the variety of reflexes like the Herring-Brewer reflexes, right? And the neural control includes the very various, the excitatory and inhibitory neurons, that I and E neurons at the level of medulla, the dorsal group of neurons, the ventral group of neurons, right? So that is the story for another day. But today what we are going to discuss is about the chemical regulation of respiration, right? So let us start this topic. The chemical regulation of respiration includes the variety of chemoreceptors, right? So chemoreceptors are the various types of receptors that detect the change in various chemicals in our blood, okay, that indirectly or directly affect our respiratory system. So there are various chemoreceptors, right? There are various chemoreceptors that are located in various parts of our body. So these chemoreceptors are located in the various parts of our body, maybe at the level of aorta, maybe at the level of our carotid arch, right? So these type, or at the level of medulla. So these various types of chemoreceptors are present in our body. These chemoreceptors detect various the concentration of various chemicals, that can be the oxygen concentration, that can be the CO2 concentration, or the level of H plus ion, and then they send a signal to the to our higher centers, whether to increase respiration or whether to decrease respiration, right? So this is what we are going to study in this chapter. For example, if there is less oxygen in your blood, then this is detected by the various chemoreceptors, right? The various chemoreceptors of our body, and they stimulate your medulla, to increase the process of respiration, to increase ventilation, right? So that your oxygen level will come up. So these various things we are going to study in this chapter. So mainly that the chemical regulation is done by the chemoreceptors. Now what are the types of chemoreceptors? So these chemoreceptors are mainly of two types, right? So these are of mainly two types. So one is known as the peripheral chemoreceptors, right? So one is peripheral chemoreceptors and the other one is known as the central chemoreceptors, right? So these peripheral chemoreceptors, these are located away from your central nervous system. That is, it is not located in the central nervous system, not located in the brain, not located in the brain stem or in the spinal cord. These are located away from the CNS, right? Whereas the central nervous system is located in the brain. These are located in the brain stem. And if you ask me what falls under the central chemoreceptor, so I'll tell you that the ventral surface of the medulla, is the example, or is the central chemoreceptor. So your central chemoreceptor, it constitutes of the ventral surface of medulla, right? The peripheral chemoreceptors, so what does it include is that it includes the various types of bodies, and these bodies are carotid bodies and aortic bodies, right? So the peripheral chemoreceptors, this includes the various types of bodies. These bodies are carotid bodies and aortic bodies. Okay, so these bodies mainly constitute the peripheral chemoreceptors. Now if you ask me where are these bodies located, so by the name itself you can understand that these aortic bodies are located in the aorta, right? So if this is your heart, right, down you have your heart, and if this is the aorta, then this part of the aorta is known as the arch of aorta, right? So you know that this is known as the arch of aorta, and in this arch of aorta there is the presence of various type of tennis racket like structures. These tennis racket like structures are none other than aortic bodies. So these aortic bodies are present at the level of aortic arch, right? Now now if you talk about the carotid bodies, these are present at the level of the carotid artery, right? So these are present at the level of aortic arteries. For example, if this is your internal carotid artery, right, this is your internal carotid artery, and this is your external carotid artery. You know that the internal carotid artery and the external carotid artery, they combine together to form the common carotid artery. So at the bifurcation point there are presence of certain tennis racket like structures, and these tennis racket like structures are none other than the carotid body. Okay, so this is none other than the carotid body. And if you ask me what is this dilated portion that is present at the level of internal carotid artery, so this is our carotid sinus, right? Carotid sinus. And at the level of carotid sinus you'll find various types of baroreceptors. So always remember that carotid sinus consists of baroreceptors, and carotid body, it consists of chemoreceptors. This is very important, right? So the peripheral chemoreceptors, it consists of carotid bodies and aortic bodies. Carotid bodies are found at the level of the bifurcation of the common carotid artery, right? So these tennis racket like shaped structures are known as carotid body that consist of chemoreceptors. And in the internal carotid artery, you'll find the carotid sinus, and these carotid sinus, they have various baroreceptors, right? Similarly, at the arch of aorta, so similarly, if this is your arch of aorta, this arch of aorta also consists of various tennis racket like structures, and since it is found at the aorta, at the arch of aorta, this structure is known as the aortic bodies, right? So this is known as the aortic bodies. And similarly, at the arch of aorta, there are presence of baroreceptors here also. So baroreceptors are present at both locations. Okay, so these aortic bodies are chemoreceptors. Do you get me? Now, in order to make this more clear, there is one drawing that is given in Guyton, right? So this is a drawing that is given in Guyton. So if you look at this drawing carefully, you will find that this is the arch of aorta, and in this arch of aorta, you will find various tennis racket like structures that are known as aortic bodies, right? Similarly, this is your common carotid artery. This common carotid artery, at the level of the bifurcation, there are presence of tennis racket like structures, and that is known as the carotid body. Now I'd like to tell you one new thing. Okay, one new thing is, what is the afferent supply to these bodies, or what are these bodies innervated by? So if you look at the innervation of these bodies, like let me show you the aortic body. So if this is the aortic body, and if you look at the nerve supply, this yellow structure, like these nerve supply, if you look at, you'll see that these are supplied by the vagus nerve. So the aortic bodies, these aortic bodies, these are innervated by the vagus nerve. So the aortic body is supplied by the vagus nerve, which is the 10th cranial nerve. Similarly, the carotid body, if you trace its nerve supply, right, if you trace its nerve supply, you will find that these carotid bodies, these are innervated by the glossopharyngeal nerve. So the carotid body, this is supplied, or this is innervated by the glossopharyngeal nerve, and this glossopharyngeal nerve is the 9th cranial nerve, right? So this is very important, right, when we discuss later about the afferent supply. So note that the aortic body, right, aortic body, these are innervated by the 10th cranial nerve. 10th cranial nerve is the vagus. It is the longest cranial nerve, and this can even extend up to your heart, right? It has a parasympathetic supply to the heart. You know that vagus is the longest nerve, it goes up to the heart. So the aortic bodies are innervated by the vagus nerve, whereas the carotid bodies, these are innervated by the 9th cranial nerve, that is the glossopharyngeal nerve. So this is our basic introduction about the chemoreceptors. So first we are going to discuss about the peripheral chemoreceptors. That means we are going to discuss in detail about the carotid bodies, aortic bodies, and how do they function. And later we are going to discuss about the central chemoreceptors that includes the ventral surface of the medulla, right? So the basic structure of the carotid body and the aortic body is the same. So let me write it under a single heading, right? Let me write it under a single heading, and the heading that we are going to make is the carotid body or the aortic body, right? Now if you look at the basic structure of the carotid body, so I have already told you that the carotid body or the aortic body are the tennis racket shaped structure. Now if you look at the cross section of this carotid body, what do you see, right? So inside of this carotid body, what are present? I'm going to draw it, right? So mainly the carotid body or the aortic body, this is mainly composed, or if you look inside the carotid body, you will mainly find two types of cells, right? So one type of cells are these cells, right? So these cells are known as type 1 glomus cells. So this is known as type 1 glomus cells, right? So this is known as the type 1 glomus cell. So if you look inside, right, if you look inside the carotid body, you will find type 1 glomus cell, right? Similarly, there is another cell, okay, there is another cell that is present over here, right? And this cell is known as the type 2 glomus cell. So basically you will understand that inside the carotid or the aortic bodies, there are the presence of glomus cells, and these glomus cells are mainly of two types. One is the type 1 glomus cell, and another is known as the type 2 glomus cell. Now if you ask me what is the function of these two cells, or which of the two cells are more important in detecting the in detecting the chemical signals, or in detecting the change in certain chemicals, then I will tell you that the type 1 glomus cell is the sensor cell. So mainly this is the cell that is going to detect the various change in chemicals, right? So this type 1 glomus cell is mainly responsible for detecting the change. So detects the change in certain chemicals, right? So mainly this cell is responsible for detecting the certain chemicals, and this cell in turn, it stimulates our respiratory centers. We'll come that in a while. This type 2 glomus cell is known as supporting cell. So mainly the function is just to support the type 1 glomus cell. So it has no major function of its own. So it is known as the supporting cells or the sustentacular cell, right? So this is known as the supporting cell or sustentacular cells, right? So this is known as the supporting cells or the sustentacular cells, right? Now I've told you that the type 1 glomus cell is mainly responsible for detecting the change in certain chemicals. Now what are these chemicals, right? So what do the type 1 glomus cell actually detect? What change do they detect? So one thing that they detect is that they detect a decrease in the arterial partial pressure of oxygen, right? So if there is a decrease in the partial pressure of oxygen in the arterial blood, that is detected by the type 1 glomus cell. So basically you understand it in this way: if the oxygen level of the blood is low, right, if there is less oxygen in blood, if there is hypoxia, that is detected by the type 1 glomus cell. So basically this means partial pressure of oxygen at the level of arterial blood. So less oxygen in arterial blood, if it is happening, then that is detected by the type 1 glomus cell. Similarly, if there is an increase in the partial pressure of carbon dioxide, if there is an increase in the partial pressure of carbon dioxide at the level of arterial blood, that we can call as hypercapnia, then that is again detected by type 1 glomus cell. So if there is hypoxia, that is detected by type 1 glomus cell. If there is hypercapnia, that is detected by type 1 glomus cell. And if there is an increase in the H plus ion concentration in blood, that means if there is acidosis, right? So if there is acidosis, that is also detected by the type 1 glomus cell. So as it can detect the change in various chemical signals, this cell is responsible. This cell is considered as the major cell, as the major sensor cell that is acting as a chemoreceptor at the level of carotid bodies, right? So type 1 glomus cell is known as the sensor cell. It detects the change in various chemicals. What are those chemicals? It is a decrease in the partial pressure of oxygen at the level of arterial blood, increase in the partial pressure of carbon dioxide at the level of arterial blood, and an increase in H plus ion concentration that we call as acidosis. Right. So now you know that the type 1 glomus cell is going to detect a change in various chemicals. Now after detecting the change, what is this glomus cell going to do? This is going to activate, or this is going to send these signals to the respiratory center, right, by the help of various neurons. So at the level of the carotid body or the aortic body, there is the presence of afferent neurons. So these afferent neurons are present, and these afferent neurons are going to carry the impulse. So these are going to carry the impulse or carry the afferent impulse to the respiratory center, and if you ask me what is the respiratory center, then I will tell you that this is the ventral group of neurons that is present at the medulla, right, at the medulla. So the ventral group of neurons, the VRG, ventral group of neurons at the level of medulla, that we call as the pre-Borginger complex. So at that level, right, these impulses are going to go, that is going to stimulate it. Okay, now what are these neurons, right? We understood that this is the afferent neurons. Now what are these afferent neurons? We have already discussed these afferent neurons can either be vagus nerve or it can either be glossopharyngeal nerve. If it is aortic body, then the afferent signals are going to go through the vagus nerve, and if it is the carotid body, then the afferent impulse are going to get carried by the glossopharyngeal nerve, right? So this is all about the carotid body. So what you have studied is that the carotid or the aortic body basic composition, basic structure is the same. It consists of two types of cells: type 1 glomus cell, type 2 glomus cell. Type 2 glomus cell is mainly the supporting cell, that is known as sustentacular cells. Type 1 glomus cell is the main cell that is responsible for the chemoreceptor function. So it is known as the sensor cell. Why is it known as sensor cell? What does it sense? So it senses the change in certain chemicals. And what are those chemicals? Decrease in the partial pressure of oxygen, and increase in the partial pressure of CO2 at the level of arterial blood, and also the increase in H plus ion concentration. So these three things are detected by the type 1 glomus cell. Now after it has detected, it is going to send the signals to the afferent neurons. These afferent neurons can either be afferent, it can be either vagus nerve or it can be glossopharyngeal nerve, depending upon the body. If it is carotid body, then glossopharyngeal nerve is going to carry the impulse. If it is aortic body, vagus nerve is going to carry the impulse to the respiratory center, because as you know that there is hypoxia, so respiratory center will have to increase the respiration, will have to cause hyperventilation, right? So this is going to get carried into the respiratory center, and which is the major respiratory center, which is the main center that is responsible for maintaining the rhythm of respiration, that is the ventral group of neurons. Guyton has said it as the dorsal group of neurons, but according to the latest research, okay, it has been found out that the ventral group of neurons are mainly responsive, these are more important, right? That is the, you know, the Borzinger complexes. Okay, so that is all about the carotid body. So did you get this? Now we are going to discuss how this type 1 glomus cell is going to get activated. We told that the type 1 glomus cell will detect the change in the chemicals. Now how does it detect the change in chemicals? How does this type 1 glomus cell detect the change in chemical, and how this type 1 glomus cell is going to send afferent into the afferent neuron? So what is the basic function, how the type 1 glomus cell works, we are going to talk about that, right? Now let me draw a basic drawing of the type 1 glomus cell. Now this is the type 1 glomus cell. Let us draw a blood vessel that is passing nearby. These are the various types of the blood vessels. These are the blood vessels, right, that is passing very close to it. Now what are the various types of signals that this type 1 glomus cell uh, detect? So one thing that it is going to detect is the decrease in the oxygen or decrease in the partial pressure of oxygen. It is going to detect an increase in the partial pressure of CO2, and it is going to detect a decrease in the it is going to detect an increase in H plus ion concentration, that is known as acidosis. So these are the major three things that are going to get detected by the glomus cell. Now how it detects, right? So this capillary has a decrease in O2, that means it is having hypoxia, right? So it is having hypoxia. Now if there is hypoxia, what is going to happen? Remember that at the level of the glomus cell, at the level of the glomus cell, there is the presence of certain type of channels, and these channels are oxygen sensitive potassium channels. So these are oxygen sensitive potassium channels. Note that at the level of type 1 glomus cell, type 1 glomus cell consists of channels, and these channels are the oxygen sensitive potassium channels. What do you mean by oxygen sensitive potassium channels? Oxygen sensitive potassium channels means that if there is enough oxygen, okay, so if there is enough oxygen, these channels will remain open, but if there is less oxygen, these channels are going to remain closed. These channels only open when oxygen is adequate. If there is less oxygen, this channel will not open. So remember it like this, okay? Now see, since there is a decrease in oxygen, right, since there is a decrease in oxygen, what will happen to this channel? These channels are not going to open because this is oxygen sensitive. Oxygen sensitive means that these channels are not going to open, so these channels will become closed. These channels will become closed. What passes through these channels? Potassium ion. Okay, potassium ion, it goes out of these channels. So since the channel is closed, now the potassium will not be able to go out of the cell. Okay, so this is very important, right? So let me write it this way. Let me write a flow chart here. Okay, so if there is hypoxia, right, so if there is hypoxia means if there is a decrease in the partial pressure of oxygen at the level of arterial blood, what is going to happen is that there is going to be closure of oxygen sensitive potassium channel. So these oxygen sensitive potassium channel will be closed. This will not open because there is no adequate oxygen. So if there is no adequate oxygen, these channels will remain closed, and if these channels remain closed, the potassium efflux will not be possible. There will be no potassium efflux. The potassium will get retained within the cell. So there will be retention of potassium within the cells, right? So within the cells there is going to be a retention of potassium. That means there is going to be high amount of positive charge within the cells. So if there is high positive charge inside the cell, what will happen to the cell? Will there be depolarization or repolarization of the cell? So you must be knowing that if there is high positive ion within the cell, there is going to be the depolarization of cell. So the cell will get depolarized, right? So since there is high amount of positive charge within the cell is going to get depolarized. Now what will happen after depolarization? Remember, it is very important. There is another channel that is present at the level of type 1 glomus cell, and that channel is known as the voltage gated calcium channels, right? So there is the presence of voltage gated calcium channel. So voltage gated calcium channel is present at the level of type 1 glomus cell. Now if there is depolarization, right, so if there is depolarization, what will happen to this calcium channel? So depolarization means there is enough voltage, so this voltage gated calcium channel will open, and if this voltage gated channel will open, there will be rapid influx of calcium inside the cells, right? So depolarization of cell, if there is depolarization of cell, there will be opening of voltage gated calcium channel. You understand the meaning of voltage gated, right? So voltage gated means there is enough depolarization, enough action potential, right? So enough impulse or the threshold has been met, right? So there is going to be an opening of the voltage gated calcium channel, and if there is opening of voltage gated calcium channel, there is going to be rapid influx of calcium within the cell. So there is going to be influx of calcium into the cell. Now what will happen? This type 1 glomus cell, it consists of various types of neurotransmitters. So there is presence of various types of neurotransmitters at the level of type 1 glomus cell. What are these neurotransmitters made up of? What are these neurotransmitters, right? So these neurotransmitters are mainly the, these are mainly the ATP, acetylcholine and dopamine, right? So what is going to happen? Please listen very carefully. So at the level of type 1 glomus cell, there are various types of neurotransmitters that are present. Now after there is influx of calcium within the type 1 glomus cell, you know that calcium is responsible for exocytosis. So there is going to be the exocytosis of the neurotransmitter. Now the neurotransmitter are going to get released out of the type 1 glomus cells, right? So there is going to be calcium mediated exocytosis, right? So there is going to be calcium mediated exocytosis of neurotransmitters, calcium mediated exocytosis of the neurotransmitters. And what are these neurotransmitters? It is very important. It is ATP, acetylcholine and dopamine. Generally the old concept was that it is mainly the dopamine was the major neurotransmitter, but if you look at the very recent edition of Guyton has mentioned that ATP, acetylcholine and dopamine are the major neurotransmitters that are released by the type 1 glomus cell. Now these type 1 glomus cell are going to generate an action potential. So here there might be like various types of neurons. So here there are different types of neurons. So at these neurons, there are presence of receptors. Okay, so there is the presence of receptors for these neurotransmitters. So these neurotransmitters are going to bind to its receptors, and after that there is going to be the generation of action potential. Action potential is going to get generated, and this action potential is going to stimulate your various the ventral group of neurons at the level of medulla, and after that your ventilation is going to get increased, right? So after what will happen, I'll tell you again. So after the release of neurotransmitters, these neurotransmitters are going to bind to its receptors, okay, into the neurons. These neurons can be either the vagus nerve, right? Vagus nerve, it can be either the vagus nerve if you are talking about the aortic bodies, or it can be the glossopharyngeal nerve if you are talking about the carotid body. So these two neurons or these two bodies are present. So after the after the neurotransmitter bind to its receptors, there is going to be production of action potential. These action potential will travel, and these are going to stimulate your ventral group of neurons present at the level of medulla, and after that the ventilation is going to get increased, right? So this is what is going to happen after there is hypoxia in your body. There is low oxygen concentration. If there is low oxygen concentration in your body, then the various peripheral chemoreceptors like the carotid body and aortic body are going to get stimulated, and in this way the various nerve impulses are going to get transmitted, and in this manner there is going to be increase in ventilation. So if there is increase in ventilation, then the oxygen level will gradually increase, right? So there is going to be exocytosis. After exocytosis, there is going to be generation of action potential. Okay, it can be the 9th neuron or 10th neuron depending whether it is aortic body, carotid body, and it is going to stimulate the ventral group of neurons, mainly the pre-Borzinger complex, and that is going to increase the ventilation. So this is all about the working of the type 1 glomus cell. Remember that this hypoxia is the direct stimulus for hyperventilation. So this is it directly influences the closure of oxygen sensitive potassium channel. Even these two signals, right, that is increase in CO2 and increase in H plus, they also cause the closure of oxygen sensitive potassium channel, but this is by some indirect method. So indirectly they are going to cause a closure of oxygen sensitive potassium channels, but hypoxia is going to directly cause the closure of oxygen sensitive potassium channels. So this is how the peripheral chemoreceptors are going to detect the various types of fluctuations in our body, and in this manner this is going to work, right? So this is all about the peripheral chemoreceptor. So we have understood how these two bodies work and how they are responsible for increasing our respiration. Now we are going to discuss about the central chemoreceptors, right? Now after the peripheral chemoreceptor, we'll talk in brief about the central chemoreceptors, right? So where is the central chemoreceptors located? So I've already told you in the beginning, these central chemoreceptors, these are located at the ventral surface of medulla. So this is located at the ventral surface of medulla. This area is also known as the chemosensitive zone. Okay, so this is also known as the chemosensitive zone, right? So the central chemoreceptors, these are located at the ventral surface of medulla, that is also called as the chemosensitive zone. Now let me talk about a brief anatomy about the ventral surface of the medulla, which will help you understand better. So this is your ventral surface of the medulla. So I'm just drawing a cross section, ventral surface of medulla. And around the medulla, around the part of the CNS, what is it filled with? What it is surrounded by? So as you know, it is surrounded by the cerebrospinal fluid, right? And this cerebrospinal fluid are obviously rich in water. As you know, it is a fluid, right? So as it is a fluid, okay, as it is a fluid, so the part surrounding it will be filled with water. Now the the medulla will obviously it is going to have blood vessels, right? So this is having blood vessels. Now what is the peculiar feature of the capillaries present at the level of CNS, at the level of brain? What is the peculiar feature of the capillary is that there is going to be presence of a blood brain barrier between the capillary and the part of the brain. So this capillary, there is the presence of a blood brain barrier. This blood brain barrier will not allow, okay, freely any substance to move in and out of the capillary. So this is known as the blood brain barrier, right? So this is known as the blood brain barrier. So blood brain barrier is present between the capillary and the CNS. So all of you know this, right? Now how these central chemoreceptors are going to influence the respiration, or how this is going to detect the change in chemical. So what is the stimulus that the central chemoreceptor is going to detect? So what is the stimulus that the central chemoreceptor are going to detect? What was the stimulus in peripheral chemoreceptors? What were the stimulus? There were three stimulus, right? That is, one is decrease in the partial pressure of oxygen, that is hypoxia, increase in partial pressure of CO2, that is hypercapnia, and increase in H plus ion concentration, that is known as acidosis. So there were these three stimulus. So these three stimulus were mainly responsible, that caused the activation of the peripheral chemoreceptors. At the peripheral chemoreceptor, one more thing, these three stimulus were responsible, okay, decrease in O2, increase in CO2, and increase in H plus ion. But where at the level of arterial blood, right? So at the level of blood vessels, okay, blood vessels. So at the level of blood vessels, if there is decrease in O2, if there is increase in CO2, and if there is an increase in H plus ion concentration, that is acidosis, then at that time what was happening, the peripheral chemoreceptors were getting stimulated. But at the central chemoreceptor, there is going to be only one stimulus, and that is the increase in the H plus ion concentration, okay, H plus ion concentration. And this H plus ion concentration is not at the level of blood vessel, but this increase in H plus ion concentration is going to be at the level of CSF. So if there is an increase in the H plus ion concentration at the level of CSF, then only the central chemoreceptors are going to get stimulated, right? So if there is increase in H plus at the level of blood vessels, at the level of arterial blood, this increase in H plus is not going to stimulate the central chemoreceptors. But at the level of CSF, if there is increase in H plus, then only it is going to stimulate the ventral surface of medulla or the central chemoreceptors. Is very important, right? Now let us discuss how this is going to take place. So for example, this is our blood vessels at the level of the blood brain, at the level of ventral surface of medulla. Now what will happen if there is a decrease in oxygen concentration? will this decrease in oxygen concentration, will it be able to stimulate these neurons? Will it be able to stimulate the central chemoreceptors? Let us consider this to be the chemoreceptor. So will the decrease in oxygen concentration be able to stimulate the chemoreceptor? The answer is no. Will this oxygen be able to cross the blood brain barrier? The answer is yes, because oxygen, CO2, these are the lipid soluble, these are the gases that are freely diffusible through the membranes, right, through the lipid membranes. So oxygen will be freely, it will able to diffuse through the blood brain barrier. But even though it is going to diffuse through the blood brain barrier, this decrease in oxygen will not be able to stimulate the central chemoreceptors, because there is no presence of oxygen sensitive potassium channels at the level of central chemoreceptors. It's very important, right? At the level of peripheral chemoreceptors, there were presence of glomus cell. These glomus cell had the presence of oxygen sensitive potassium channels. That is why a decrease in oxygen was a stimulus for the peripheral chemoreceptor. But at the level of central chemoreceptor, there is no oxygen sensitive potassium channel, so a decrease in oxygen concentration will not be able to stimulate the central chemoreceptors, right? So decrease in oxygen concentration is never a stimulus to the central chemoreceptors, right? Now, will an increase in H plus ion concentration at the level of blood vessels be a stimulus to the central chemoreceptors? The answer is no. The answer is no. Okay. So, but here you have written an increase in oxygen concentration, but I've written the increase in H plus ion concentration at the level of CSF. So, at the level of CSF, if the hydrogen is high, then that is going to be a stimulus for the central chemoreceptors. But at the level of blood vessels, right? This is the blood vessel. So, at the level of blood vessel, if the H plus ion concentration is high, this will not be a stimulus for the central chemoreceptors because of the blood brain barrier. This H plus ion is a positively charged ion, and ions can never cross the blood brain barrier. Okay, so ions can never cross the blood brain barrier. So this H plus ion concentration will also not be a stimulus for the central chemoreceptors. Now, how will the central chemoreceptor increase? So is there any role of CO2? The answer is yes. So what will happen if there is an increase in CO2 at the level of arterial blood? Remember, will CO2 be able to diffuse through the blood brain barrier? The answer is yes, because it is a gas. So as it is a gas, this CO2 can easily diffuse through the blood brain barrier, and this can come into the level of ventral surface, I mean of medulla, right? So it can come here. So after that, this CO2 can easily pass into the CSF. Okay, CO2 is a lipid soluble, it is a gas, it can easily diffuse through the lipid membrane. So this can easily cross the blood brain barrier, and it can easily cross and reach the CSF. Do you agree or not? Yes. So now what will happen to this CO2 at the level of CSF? Remember that as I've told you, this CSF is very high in water. So this CO2 is going to combine with water. So what will happen if CO2 combines with water? This is going to form carbonic acid, right? So this CO2, if it combines with water, this is going to form the carbonic acid, and this carbonic acid will be splitted into H plus and HCO3 minus. You already know this, right? So H2CO3 will again split into H plus and HCO3. So as you can see that there is now the presence of H plus ion at the level of CSF. Now if there is H plus ion at the level of CSF, can you tell me whether this will be the stimulus for the central chemoreceptors or not? The answer is yes. The answer is obviously this H plus is going to be a stimulus for the central chemoreceptor, because we have already written here that increase in H plus ion concentration at the level of CSF will always be a stimulus for the central chemoreceptor. So in this manner, if there is an increase in CO2 level at the level of arterial blood, then this CO2 will produce carbonic acid at CSF, and this carbonic acid will dissociate to form H plus ion at the CSF, and this H plus ion will be a stimulus for the central chemoreceptor. So in this manner, the central chemoreceptors are going to get activated, right? So it is a very important concept, what is happening to oxygen, what is happening to H plus, and what is happening at the level of CO2. So this thing we are going to write it, okay? So first is hypoxia. Hypoxia, what do you mean by hypoxia? So hypoxia means a decrease in O2 concentration. So will this decrease in oxygen concentration be a stimulus for the central chemoreceptor? The answer is no, right? So this oxygen concentration is never a stimulus. So, just a brief summary. Hypoxia, that is decrease in O2, is never a stimulus for central chemoreceptor because there is no presence of any oxygen sensitive channels, right? So there is no presence of any types of oxygen sensitive channels. So hypoxia is only a stimulus for the peripheral chemoreceptors. The same thing with acidosis. It is not a stimulus for central chemoreceptor because the ionic molecule, it cannot cross the blood brain barrier, so it is also only a stimulus for peripheral chemoreceptors. Now if you talk about increase in CO2, this increase in CO2 can cross the blood brain barrier and it can reach the CSF. So at the CSF, this CO2 will be reacting with water to form H2CO3, and that is going to dissociate to form H plus. So since there is an increase in H plus at the level of CSF, it is able to stimulate the chemoreceptor. So hypercapnia is the only stimulus that is able to stimulate, that is able to send stimulus to both central and peripheral chemoreceptor, and that is the reason why we tell that CO2 is a more stronger stimulus for the ventilation, right, then as compared to O2. So an increase in CO2 is a more stronger stimulus for respiration than the decrease in oxygen, right? So that is all about the our chemoreceptor or the chemical control of respiration. Thank you.